Good morning, everyone. In today's class, we will try to understand an important subunit in the course Analytical Biochemistry 2. This is about different types of models that are used in biological research. So, this concept is more of research oriented, and we will try to understand the in and out of the scope and relevance of model systems in biological research, and why a model system is required, what is the criteria for a model biological system, and what is the outcome of using a model system in understanding the process and concepts of biological research. So, all these questions will be answered when we try to understand about the model systems used in the biological research. So, as you know, there is a development of wide variety of species which has been used for various studies at one time or another, and a relatively small number of organisms has provided most of our knowledge about the developmental process or the mechanisms, right? So we can consider these relatively small number of organisms as models for understanding the process involved. Now, a model organism, when we say an organism as a model organism is a non-human species which we use extensively to understand the process and concepts. That means a very specific process and concepts of biological system, right? So these organisms, they are chosen because it is believed that the discoveries made in the model organisms will surely provide a insight into the working of other organisms, and this include humans. Say for example, sea urchins and the frogs, they were the main animals that those were used for the first experimental investigations at the beginning of the studies using animals. Because of their developing embryos, they are easy to obtain. And in the case of frog, they're sufficiently large and they are robust for relatively easy experimental manipulation, even at later stages. So, what is need to be focused here is these organisms, be it the frogs or sea urchins. Now you have a list of a wide variety of organisms. When they are chosen, it is believed that whatever the data you get or whatever the discovery you make will surely provide an insight into the working of other organisms, including the humans. And among vertebrate frog, xenopus, mouse, chick, and you might have heard about the zebra fish among vertebrates. I am speaking about vertebrates. You have frog, xenopus, mouse, chick, zebra fish. So these are taken into consideration for model system. And in among invertebrates, we have the Drosophila, the nematode worm, that is C. elegans, so these organisms are presently the focus of most attention for the reason that they are well studied developmental genetics and they can also be modified at genetic level, right? So that's the reason. So one thing what you should remember is the model organisms are chosen for the reason that they will provide information more accurately so that that can be translated to humans. Most of the time this is true about using a model organisms. And the reasons for selecting them as model system, they are partly historical. Once a certain amount of research has been put on one animal, so it is more efficient to continue to study rather than starting from the beginning again with another species. So it is always better that you perform some experiment using some model system and then further studies should be continued using the same model system rather than start at the beginning again with another species and partially a question of ease of study and biological interest. So each species has its advantages and disadvantages as a developmental model. Say for example, in case of the chick embryo, it has been studied as an example of vertebrate because fertile eggs are easily available. The embryo withstands experimental microsurgical manipulations very well and it can be cultured outside the egg. However, a disadvantage is that very little is known about the genetics as compared to that of the mouse. So, each species has its own advantages and disadvantages. right? In case of chick embryo, the disadvantage is that a little is known about the genetics as compared to that of the mouse. Now, although the mouse is more difficult to study in some ways, as developmental mutations have been identified in the mouse, so it is amenable to genetic modifications by various transgenic techniques. And it is also the best experimental model for studying the mammalian development, including humans. For the reason that genetically the human and the mouse are very similar. With many of the disease related genes, they are approximately identical. So that is the reason why it is supposed to be the best experimental model that is used for studying mammalian development. So that includes humans too. Now we have the zebrafish. So this zebrafish is recently added to the list of vertebrate model systems because it is easy to breed in large numbers in comparison to the mouse, and the embryos are transparent, that makes the experimental design more easy. And so cell divisions and tissue movements can be easily followed, that means you can do it visually, and it has great potential for genetic investigations. So as I said, each species has its own advantage and disadvantage. Now if you look at zebrafish as a model system, which has been added in the list of vertebrate model system, it is easy to breed in large numbers. Embryos are transparent so that the cell divisions and tissue movements can be followed visually, and it has great potential for genetic investigations. And we know that the study of genetics of the fruit fly, that is Drosophila melanogaster, it has been studied since the beginning of this century. That is from 20th century itself. So research on its development has come into light only in relatively recent times with the advent of molecular biological techniques. So earlier it was not that much. Now you have lot of information on the development of Drosophila melanogaster, and it has made possible with the advent of molecular biological techniques. So studying Drosophila melanogaster as a model developmental system is quite recent. Due to simple morphology and less requirement of space and the cost, the expense. So these fruit flies, that is the Drosophila melanogaster, they are most studied organisms in biological system. So we have seen why the chick embryo is chosen and what are the advantages of zebrafish and what is the scope of using Drosophila melanogaster. So with this prelude, we'll try to see what are the criteria for a model biological system. Number one, the organism used should be easy to maintain. Number two, the operational size should be small or it should be convenient to handle. Number three, the experimental protocol should be inexpensive to maintain the model system. Number four, The model organism should have short life cycle. So whatever the manipulation that is done can be easily studied if the life cycle is short. Number 5, the organism used should be, or you need to study about genetics, then it should be capable enough to manipulate genetically. Number 6, it should be potential to deliver economically important results. So these are the important criteria for a model biological system. Number 1, the organism used should be easy to maintain. Number 2, its operational size should be convenient. Number 3, it should be inexpensive to maintain. Number 4, the model organism used should have short life cycle. Number 5, the organism used can be genetically manipulated. And the last one, it should be potential to deliver economically important results. So by comparing the developmental mechanisms in a variety of organisms, so this becomes possible to identify that mechanisms among groups of organisms. So comparative studies suggest that if there are differences in the details, so it is likely that most basic mechanisms of development, they are similar in on all animals and are derived from the earliest animal ancestors. So most of the times it is similar in almost all animals. So thus, if you elucidate the developmental process in one animal, is surely of great help in understanding the developmental process of another animal. So most of the time this is true when your research is all about the developmental biology. Most of the time the animal models are chosen to understand the developmental process and concepts, right? And there is a quote on the relevance of animal models to biological research. The quote is, research on relevant, carefully designed, well characterized and controlled animal models will remain for a long time an essential step for fundamental discoveries, for testing hypotheses at the organism level, and for the validation of human data. Unquote. So what does this mean? I wanted you to understand. That's the reason why I have used this quote. This quote will tell you the scope and importance or the relevance of use of the model system, especially the animal model system. It says this kind of approach, that is the animal models, will remain for a long time an essential step for the fundamental discoveries. And if you have an hypothesis, you can test the hypothesis, and also for validation of the data that has been obtained from various research. so that it can be translated to the humans. Right? So this is beautifully said. So whatever the research, you do it on well characterized, carefully designed and controlled animal model system, definitely it will remain as an essential step for the fundamental discoveries, to test the hypothesis and for the validation of the data. So the use of animals for scientific purposes is both a long standing practice in biological research and as well as medicine. And the remarkable anatomical and the physiological similarities between humans and animals, particularly the mammals, they have prompted the researchers to investigate a large range of mechanisms and assess novel therapies in animal models. So why it is so much required? Because of remarkable anatomical and physiological similarities. So that has prompted many of us to investigate a large number of novel therapies against various diseases in animal models. So these can be done before applying the data or translating the data to humans. So you need not directly test the hypothesis or do the fundamental discoveries on humans. It's not possible. So you have a model system wherein the model system is anatomically and physiologically similar to the humans, especially in case of mammals, so that you can then translate the data, whatever the data you get from animal model studies, directly you can translate it into the humans. But however, not all results or the data obtained on animals can be directly translated to humans. It's not possible. Right? And at the same time, the place of the animals in our modern societies is often debated. Now, the ethical issues, particularly the right to use animals to benefit human purposes. How good it is to use the animals only for the benefit of the humans. This is the debate. For the reason that there is a possibility that the animals are harmed. And you are doing all this only for the benefit of the human purposes. How good it is. So, the place of the animals in our modern societies is seeing a lot of debate. So, for this we have down the lane lot of policies that have come in. So, we will discuss about it little later. Right? The humans and other mammals are very complex organisms, as we all know. And we know that the organs achieve distinctive physiological functions in a highly integrated and regulated manner. So the relationships involve a complex network of the hormones, circulating factors and cells, and a crosstalk between the cells in all the compartments. So whenever a biologist look at the organism, he or she will try to dissect at multiple levels. Number one, at the level of molecules. Number two, cells. Number three, organs. Number four, physiological functions, both in case of the healthy and diseased conditions. So we have the studies at molecules, cells, organs and physiological functions in healthy or the disease conditions. So all these levels of studies or investigations are required to get a full description and understanding of the mechanisms. The first two, and in some instance the third one, the level of organizations can be studied using in vitro approaches. See, if you want to understand about the molecules, the cells, or even the organs, sometimes. So there is an alternative approach, that is the in vitro approaches, that is through cell culture. So these techniques have become very sophisticated to mimic the three-dimensional and complex structures of the tissues. And these alternatives, that is in vitro approaches, nowadays they represent major scientific advances and they have replaced the use of animals. But on the other hand, number 4, the exploration of physiological functions and systemic interactions between organs requires a whole organism. So here you cannot have the in vitro approach. For example, in the case for most hormonal regulations, for the dissemination of microorganisms during infectious disease, or for the influence of the intestinal microorganisms on immune defense, or in case of the development of brain functions, you cannot have the in vitro approaches. So in these cases, no in vitro model is available. Of course, there are simulations available, but it does not translate the research simulation data into the actual research. So no in vitro model is currently available to fully recapitulate these interactions and investigations or studies on humans and animals. That's the reason why animal models are still necessary. Hypotheses and models can emerge from in vitro studies, but they must be tested and validated in whole organism. Is that clear? So if you have hypothesis, you can do it in vitro model system. But the hypothesis, when it comes for testing and validation, it has to be in a whole organism. Because we are very far from being able to predict the functioning of a complex organism from the study of separate cells, tissues, and organs. So what we do in tissue culture, it is all separate. Cells are separate, the tissues are separate, the organs are separate. There is no crosstalk of these complex factors. And we are still very far from being able to predict the function of such a complex network. So you require a whole organism in order to test and validate whatever the research hypothesis you have proposed. Right? So what is understood is that animal models have been used to address a variety of scientific questions, be it from basic science to the development and assessment of novel therapeutic approaches. And the use of animals is not only based on the vast common factors or commonalities in the biology of most mammals, but also on the fact that human diseases often affect other animal species. So whatever the disease pattern you see in humans most of the time is also observed in animal species. So it is particular the case for most infectious disease, but also for very common conditions, say for example type 1 diabetes, hypertension, allergies, cancer, epilepsy, myopathies, and so on. Not only are these diseases shared, but the mechanism are often similar to an extent of 90%. Right? So most of the veterinary drugs they are used to treat animals, they are identical or they are very similar to those used to treat humans. Is that clear? So that is a reason why you require the whole organism for testing and validation of the data. Right? So I'll stop my discussion over here. We can continue our discussion in the next session. Thank you very much.